a new window into the brain opened. Scientists have been struggling to develop a way to monitor live human cells and their connections in the brain. Researchers from Imperial College London, in collaboration with a group from the University of Cambridge, opened up a new technique to look into human brain cells alive. They transplanted human brain cells into a mouse brain, and for the first time watched how they grew and connected to each other. This allowed the team to study the way human brain cells interact in a more natural environment than previously possible. The team used the technique to model Down syndrome, using cells donated by two individuals with the condition. The scientists say their approach could be used to study a range of brain conditions in the future, including schizophrenia, dementia or autism. The study was published in the journal Science on the 11th of October 2018. It describes how researchers saw differences in the brain cells from the individuals with Down syndrome compared to brain cells from a person without the condition. Dr. Vincenzo Di Paola, from Imperial's Institute of Clinical Sciences, is the lead author of the study. Although some of the connections formed between the brain cells from the individuals with Down syndrome were more stable and abundant, they communicated in a slightly less coordinated fashion. The results suggest the reduced coordinated activity and increased stability of connections in Down syndrome may be linked to cognitive function. Professor Rick Livesey, joint co-corresponding author from the University of Cambridge's Wellcome Cancer Research UK Kurgan Institute, commented, "Working together with the Imperial team has allowed us to extend our previous work on making stem cells and nerve cells from people with Down syndrome, to study how those nerve cells develop and function when put in a living brain. We are very excited by how much we have learned and the new avenues this has opened up for understanding Down syndrome." Dr. Raquel Reel, a neurologist from Dr. Di Paola's group at Imperial College London and joint first author of this study, added, "The transplantation of human brain cells has allowed us to monitor their maturation over time. Ultimately, we detected that cells from Down syndrome individuals are not as active as normal cells at a crucial stage in their development, and this could have important implications for some of the symptoms of this condition." Crucially, the technique allows scientists to study how brain cells communicate. The connections between brain cells, which enable them to talk to each other, is often the first thing to be damaged in conditions such as dementia and Parkinson's. This happens long before the brain cells themselves start to die. But the connections are so tiny, that no type of scanning tool available, such as MRI or PET scans, can see them. Researchers used a revolutionary microscopy technique, called in vivo two-photon microscopy, which allowed them to see not only individual live brain cells, but also the connections between them. This new imaging approach may have taken them one step closer to this. In the study, they created human brain cells by reverse engineering skin cells. This process involved taking a few skin cells from volunteers with Down syndrome, and then reprogramming them in the lab to form brain cells. They then engineered those neurons so their activity could be monitored. They implant these human neurons in the brain of live mice and monitored their development and function over time. The human brain cells not only formed complex networks, but also started communicating in a way that was very similar to normal brain cells. They expected only a few of the human brain cells would grow within the mouse brain. To their surprise, the human brain cells thrive, and soon talk and work together. It is still not clear to what extent the transplanted human brain cells resemble the organization and complexity of their counterparts in the human brain. The Imperial team now hope to refine this technique, and potentially use this approach to study other neurological conditions also. Thank you.